All right, for some reason I'm sitting in my classroom and there's absolutely no students, but since class is in session, I plan on holding it. So I'm just going to finish up and talk to myself and start with what we ended off with on the last class that we met. I'm talking to myself. Come on in. It's me and you today, I guess. All right, so we're picking up with the immune system. I don't know where everybody is. Yeah, sit up here, we'll talk. Maybe they thought if nobody showed up to class that we would push the exam off. I don't know, but they were sadly mistaken, because I don't mind talking to myself or just talking to you. That'll be fine. All right, so we're going to pick up with, we're on page 775 in the textbook, picking up with adaptive immunity and overview. So this is figure 21.8 on page 775. We finished talking about that last class. We talked a little bit about lymphocytes, where they were born, bone marrow. And we have two different types, B cells, T cells, and we also have something called antigen-presenting cells, which are extremely important for the whole immune response. So this diagram is important because you have to know where they get their education or become what we call immunocompetent. So the two different types of cells, B cells and T cells, are both born in the bone marrow, but T cells actually migrate to the thymus, and that's where they become immunocompetent.
So does that change when we get older and our thymus shrink?
Correct. Okay, so we don't have as many. Mm-hmm. We pretty much have everybody set in the, when you look at this diagram, you see step 3, seeding the secondary lymphoid organs in circulation. That's pretty much been done when we're young. So we're going to continue to make these cells, but as far as getting them their education goes and putting them in place, that's pretty much done in our early years. That's why the thymus gets smaller and smaller and smaller as we age. So know the difference between T cells, B cells, and where they become immunocompetent. And that's basically outlined in this diagram. Also, when antigens are encountered by these cells, they become activated. Then they're going to proliferate and differentiate. So that's also included in this diagram as well. Any questions on that? No? Good. All right. So, where the heck is mine? Where the heck? You see the arrow?
No.
I know. Don't see it either. Oh, there we go. There we go. All right. So, lymphocyte development, maturation, activation. Immunocompetence is basically the lymphocyte's ability to recognize different antigens. And that comes from proteins generated by your genetic material. So there's actual receptors on these cells. And this allows them to become immunocompetent, to be able to recognize antigens in their environment. This diagram tells us how that education takes place. And it takes place with the help of a very specific protein on the surface of these cells called MHC. And that is your major histocompatibility protein. Sometimes you've heard them referred to as self-antigens. This is on the surface of these cells to help them become immunocompetent and for other cells to recognize them as self as well. So a couple of different processes, positive and negative selection, takes place. In this diagram, it's pretty well lined out for you. These specific proteins with a positive, it's a two-step process, positive and negative selection, in order to pass the test, I say, in order to survive, to become part of your immune system cells. You make a lot of these cells, T cells specifically, only 2% of them pass the test. If they don't pass this positive and negative selection, they're going to,
Commit self-suicide.
yeah, they're going to undergo self-suicide, apoptosis.
So are you going to want us to know, like, because in the AMPHS class, like, as we're talking about this, I'm seeing the diagram in my head. She drew us a little picture, and like, are we going to need to know that for, like, the essay questions, stuff like that? Like, that they connect, and then the T-cells excrete or something like that. In my mind, it's very clear, but the words are...
You mean as far as positive and negative selection goes with MHC and its involvement? Yes, you need to understand that for sure. And the nice thing is this diagram is pretty self-explanatory. So positive selection, if MHCs don't line up properly, in other words, the cell can't recognize its own MHC, cell death. If they can recognize MHC for self, they pass the first test, that positive selection test. The next step, and again, this is for T cells, only because we understand T cells better and how their education goes about in the immune system.
Mm-hmm.
We're still a little sketchy on what goes on with B cells. We think it's something very similar, but we know for sure what goes on with T cells. Second step, negative selection. So if the fit is too tight, in other words, I'm going to fit in to my self antigens and not let go, which will then lead to me killing my own cells, cell death.
Yep.
If
It's a really cool word.
exactly. So again, negative selection here is I recognize my self antigen, but I don't stick on so tightly that I'm going to actually destroy the cell. Then I pass the test, then I go on and I'm allowed to seed all of the secondary lymphoid organs. So this is where in last class I talked about, some fictional numbers. Say there was one cell in every single lymph node that was able to recognize a specific antigen. That's the seeding process. Once activation takes place, we're going to see that a lot more of those cells actually are produced. So understand what self-antigens are, what those MHC complexes are, and understand the process of positive and negative selection. Then go into seeding, and then in order for an immune response to take place, these cells, which are seeded, we call naive, have to become activated. This is a neat picture on page 777, figure 2110 of a dendritic cell. And these guys are the ones that are going to help other cells recognize foreign antigens. You see them under antigen-presenting cell description. So the book then goes on to talk about we talked about naive cells, then we talk about cells that are stimulated. So antigen encounter and activation in your textbook on page 776, 777 talks about what happens to the cell when antigens come in contact with them, or they come in contact with the antigens. They're going to start to proliferate
This is what I'm saying.
and clone themselves. Yeah. So yes, you have to know this. Figure 21.11 on page 778. So when they come in contact, they become activated, and they're going to go into attack mode. They clone themselves, and that describes clonal selection at the top of page 777. So the cells will basically make more of themselves, and they'll break off into two categories. If you look at this diagram, we happen to see a B cell. B cells have on the surface of them substances that are very similar in formation to antibodies. And when specific antigens come in contact with them, it's going to stimulate them. They're going to start making more of themselves. And they're going to break off into two groups. One group is going to become plasma cells. And those cells are going to actively produce more of the same antibody that we saw on the surface to begin with. The other group of cells are going to become what we call memory cells. So these guys are going to lay in wait just in case this antigen comes back. Those memory cells, again, my fictional numbers, we're going to see instead of one cell now in every lymph node that can recognize the antigen, after that stimulation has happened, we're going to see 10 cells.
And isn't this why vaccinations work?
Correct.
Because they introduce the virus or whatever.
Exactly.
To the body, and then the B cells remember.
Exactly. Because they undergo this response, now they're going to have 10 times more cells in weight for that specific antigen. That way, and we'll see in a minute that diagram that you see on page 779, telling us the numbers of antibodies in circulation versus first exposure and second exposure. We see the second time you become exposed to a specific antigen, you have the ability to very quickly make 200 times more antibody than you need to neutralize those antigens. That's why they don't make you sick the second time. So we understand that proliferation, once they're stimulated, form two subsets of cells. In the case of B cells, we see plasma cells that are going to make antibodies, and the numbers of those antibodies will increase in plasma, and then the memory cells. And the memory cells are going to allow us to have a very rapid response to a second exposure to an antigen. So this diagram here, or table 21.3 on page 779, gives us a nice breakdown of some of the different differences between B cells and T cells. So you should know the difference between a B cell and T cell. The book then goes on to talk about humoral immune response. And basically humors are fluids that flow through the body. So we're talking about antibodies in those fluids when we talk about humoral response. Before I go on too far, I want you to understand the different types of cells that fall under the category of antigen-presenting cell. Basically what an antigen-presenting cell does is it uses those MHC proteins. And we see that there's actually two different types of MHC, MHC class 1 and MHC class 2. Not only are they recognition proteins, but they're also going to help other cells recognize antigens that an immune response is going to help fight off. And that's what these guys do. They actually engulf particles and present the antigen of interest on those MHCs on their cellular surface. So some of the different types of antigen-presenting cells are some of those dendritic cells that we saw in that neat little diagram before. Some different macrophages can do that too. And some of the macrophages we see in tissue can do this, and also certain types of T cells can do this as well. In your textbook, they talk about effector T cells. And then some B lymphocytes can act as antigen-presenting as well. And again, these cells are basically going to show the rest of the immune system what they're after. When I use my antigen analogy, basically they're going to destroy the invader, take the antigen determinant, in the case of antigen it would be her head, and stick it out for all the rest of the cells to see.
Is this why, things like lupus come up where they
That's more of an
identify
autoimmune.
Yeah, where they identify like a healthy cell as an invader.
Correct. Because they sort of got by there or altered themselves in ways so that they can get by what they learned when they become immunocompetent. Okay? So the system sort of fails when we have an autoimmune disease. So they no longer recognize self as self. So, all right, then antibody production under humoral immune response, activation and differentiation of B cells. So this is where we create what we call our immunological memory. Once we've come in contact with something foreign, and we saw it in that previous diagram on page 778, we create memory cells that can recognize and act much more quickly than the few cells we had before we had our first encounter. This diagram is a great example, visual example of how much more antibody can make and how much faster. So think about chickenpox. You know, when I was a kid, everybody had the chickenpox. Now everybody gets vaccinated. But when we had the chickenpox, it was usually about 10 days before we knew we had the chickenpox. And unfortunately, we would have done what to all the other kids in the neighborhood? Yeah, infected everybody else, because it took that long for the virus to build up enough in our system to cause problems. About 10 days is when we start making enough antibodies to start neutralizing that virus. But unfortunately, all those little pustules start popping out and we're infecting everybody. So if we look at immune response, first exposure to antigen, say chickenpox is antigen A, when we have our first exposure, it takes our bodies almost 6 days to even start producing quantities of antibodies that will even start neutralizing. So about 7 to 10 days, we're going to start to see a peak in those antibodies. That's when we build up enough neutralizing antibodies to start neutralizing foreign invader. The second time we come in contact with chickenpox, so we get the chickenpox, we get sick, we get better, we're all great. The second time we come in contact with chickenpox, at that day 28 in this diagram, we're going to make hundreds more antibodies. So 10 to the first antibody titer is enough to take care of chickenpox. We're going to make almost 10 to the fourth antibodies in circulation. That virus is going to get a chance to build up in our systems. It's not going to get a chance to make us sick. So, will we get chickenpox again? No, because the virus will never be able to build up enough to make us sick like it did originally. Again, this is a normal response. Unfortunately, sometimes this doesn't work so efficiently. So sometimes we do get this disease again. Or we never made enough antibodies or memory cells in the first place to allow the second normal response to happen. So that happens. But that's our immunological memory and our response to antigens that have come into our system. So when we talk about eliciting an immune response, this is what I'm talking about. Now, there's different ways to elicit an immune response or to acquire humoral immunity. We can do it actively, and that's basically getting sick. Or we can do it passively, introducing that antigenic determinant to our patient, having them undergo this whole process of an immune response, but because the antigen we introduce to the patient is not active, it won't make them sick. So, you should understand the differences between active and passive immunity. So naturally acquired immunity is me getting the chickenpox in the neighborhood, getting the chickenpox, undergoing an immune response, making enough antibodies, making enough memory cells. We can also actively do this by giving people vaccines. So they're the actual antigens, but they have altered them so they're not dangerous or pathogenic. In the diagram, we see the term attenuated, and that's basically what attenuated means. So they're dead or inactivated. They can't make us sick, but they still possess all of the antigens that our cells would recognize if they actually were alive. Then we can passively do this. We can naturally acquire passively antibodies via mother-fetus interactions. So across the placenta, in mother's milk, mom can give me antibodies and help me fight off any foreign invaders that might come in. We could also take antibodies and inject them into you. One of the most famous artificially acquired passive immunities is something called gamma globulin. Another one you might be familiar with, and we talked about this when we talked about blood typing, was the RhoGAM shot. Remember we talked about anti-D antibodies and how my body won't make them unless what? No, unless you come in contact with the antigen. So if I'm an Rh negative mother, and just dust off some brain cells, you might remember this discussion, I don't have antibodies to the Rh antigen until they're introduced into my body during that childbirth process. If I can give you enough antibodies to neutralize the antigens that come into your system, you're never going to undergo an immune response. So if I, with a RhoGAM shot, can give you all of the antibodies you need to neutralize any antigens that come into your system, you won't undergo an immune response. But I'll still neutralize the antigens. Okay? Does that make sense? Yes? No? Maybe? Kinda? Sorta? Yes? See, now you're under the gun because nobody's in here, so you have to answer me. So know the difference between active and passive antibody production or humoral immunity. Next thing the book talks about is the basic structure of an antibody. And the reason you always see it as little Ys is because that's what it's actually shaped like. So antibodies are made up of very similar structures that look like these little Ys, called antibody monomers. So in this diagram that you see on page 781, figure 21.14, we see an antibody monomer with all of its very similar parts. So we're going to talk in a minute about all the different types of antibodies we make, but they all have these basic parts. So they all have the basic structure that is what we call the stem region. And then they have a bend and two places in what we call the hinge region. They have a heavy chain, they have a light chain. And in that Y, those little bends on the ends, this is where we see differences from one antibody and the next. So if you look at the diagram, we see like a purple and a bluish color at that region. This is where we see differences. This is what makes one antibody different from another. It's called the antigen binding site. So know that all antibodies are made of these antibody monomers and basically held together by little bonds called disulfide bonds, consisting of a stem region and a hinge region, and then the differences in the antigen binding site. So there's different types of antibodies, and on antibodies, and it's too bad they took this away, there's a couple of different binding sites for some of the other players in an immune response. On antibodies, there's binding sites for macrophages. My brain just went dead. Phagocytic cells. Because antibodies are actually, the only thing they're going to do are neutralize antigens. And then who's going to get rid of the antigen-antibody complex? Phagocytes. Okay, so they're basically going to neutralize antigens. That's all they can do. The phagocytes are the ones that are going to destroy the complexes and get rid of them. Do we have class at 12:30 or 12 o'clock?
12:30.
Whoa. That's why I'm like, you're going at it, and I'm like, I don't know if you were just answering questions or.
I was just saying, I feel like I've been here forever, so.
Oh, well. Well, I started anyway.
Oh, you did start?
Yeah. You can catch up on the thing. I know. Well, that's what I'm wondering. I come in here, there's nobody here. I'm rushing.
How long have you started?
Since 12 o'clock.
12?
No.
Yep. Oh, yeah.
You have
Huh?
Where have we been?
Well, anyway, listen to the video and you'll get the beginning of the conversation. Yeah, well, that'll be your busy studying part. Oh my gosh, I'm losing my mind. So, I thought nobody showed up to class.
I was right there with you.
I know you were.
You were really lecturing for a half an hour?
Yeah.
Yeah, I was the only one in here. I seriously thought I was the only one that was going to be here.
Pause, rewind.
No. You could go rewind. I'm not going to say it again.
Did we get out of class a half hour early?
No. You can get out of class a half an hour. They're going to stay.
I was studying with Dr. C, so we technically...
Holy moly, I'm losing my mind. All right, so antibody monomers, yes? All antibodies are made of these monomers, so understand this structure. Again, it's figure 21.14 on page 781. Now, there's different classes of antibodies. Remember MAG. What's MAG?
IgA, IgG, IgG, IgG.
You got it, MAG. IgG. Why are they IG? IG. What are we calling these? Immunoglobulins. So these are our antibody proteins. So the different classes are IgM, IgA, IgD, IgG, and IgE. Again, the Ig stands for immunoglobulin. And these different classes are produced at different times and secreted at different times in different amounts in different places. But basically what an antibody does can be seen in this diagram here on page 782, figure 21.15. And this is their mechanism of action. This is how they work. They don't destroy cells. All they do is mark them and neutralize them. So when foreign substances enter the body, say that foreign substance is that complex, a virus that we see over there in that diagram, or it's cells, or it's some sort of particulate. Antibodies are going to do a couple of things. They can either neutralize them, surround them so they can't be harmful. They can also attach to several different cells and cause them to fall out of solution. That's called agglutination. You saw that when you did what in lab? Blood typing. You took antibodies to a specific antigen. So if you were type A blood and you put anti-A antibodies on your blood, what happened? Clumps, agglutination, and that's why. So it caused a bunch of cells to clump together and agglutinate. It can also do the same thing to particulates and cause them to come out of solution. And these are markers for the other cells in immune response to come and get rid of them. Who are the other cells in an immune response? Who's going to get rid of these complexes?
Macrophages?
Yeah, macrophages, phagocytes. They could be neutrophils. They can be macrophages. Those phagocytic cells are going to help get rid of these complexes. They're also going to help enhance certain things when we form complexes that antibodies help us do. So they can fix and activate things like complement. What was complement?
It's a chemical.
It's 20 different proteins, right? That when they become activated, what are they going to do?
They're going to make a MAC
Yep, MAC.
complex.
Exactly. They're going to form that. What's a MAC stand for? Membrane attack complex. Exactly. And what's going to help activate them when antibodies do what? When they're fixed to cell surfaces. It's going to help activate complement. Complement can then come into that cell and do what?
Please.
Poke a hole in it, cause it to burst, so it can lead to lysis. So fixing and activating other cells in an immune response is another job of what antibodies are there to do. So they can also enhance inflammatory response. Inflammatory response is going to call cells to an area and cause them to be on board to help fight off foreign substances. Things like neutrophils, when we get a cut, one of the messages that are sent is going to help call them to the area. What was the word for that call to the area of cells? Chemotaxis. Okay? And that's one of the things antibodies are going to help us do.
Can you just explain a little bit in more details about how those antibodies actually activate complement?
When they become fixed to an antigen on the surface of a cell, there's actually a group of proteins, when they sense this complex, that will activate them and bring them together.
And that's what activates
Turns them on. Causes them to band together. Okay? And that complex on the surface of cells is what helps them band together. Okay? So, understand why or how antibodies do their thing. They can activate, neutralize, agglutinate, precipitate, enhance phagocytosis. They can fix and activate complement, which can lead to lysis. The other thing you need to know, and you're going to hate me for this, but you'll get over it, is the different classes of immunoglobulin. I want you to know their structure. They all are made up of antibody monomers, similar to the diagram we saw previous, but some of them are made of more than one of these monomers. For example, IgM is one of the largest antibody complexes we make. It's called a pentamer. So how many of the monomers make up IgM? 5. So there's 5 different groups of monomers that band together to form IgM. This is typically the first immunoglobulin we see in a primary immune response. So we pump out IgM. The most effective, though, is not IgM. Even though it's got 10 binding sites for antigens, you think it would be the most effective, but unfortunately it's kind of cumbersome because it's so big. The one we're going to see the most abundant in our circulation and the most effective of the Ig classes is IgG. He's not on that, but he's in here. IgA is another one. So again, know what it looks like and know where it exists. Okay, when do we see IgM?
different secretions.
Yep, we're going to see it in different secretions in the body, and again, it's one of the first ones on site. IgA, the second one, is a dimer. What's that mean?
2.
Yep, 2 antibody monomers. We're going to see it on things like epithelial cell surfaces, mucous membranes, in the epidermis. What's the epidermis?
The outer layer of the skin.
Outer layer of the skin, exactly. Plasma is typically where we see this guy. The next guy.
You want to know if they're, like, you want to know that it's a pentamer, a dimer, or a monomer.
Correct. And then where are you going to find it? When would you see it? Where, in which secretions, in which instances? And again, it's all here in this diagram for you. IgD is another monomer found on the surface of B cells. So IgD type antibodies or immunoglobulins are what we see on the surface of B cells that come in contact with antigens that activate B cells.
So they're not free in the plasma or something. They're usually just on the B cells.
Typically, yeah,
Binding-
binding to the B cell.
Is it
On,
binding site?
no, it's on the surface of the cells, and that's what antigens bind to activate
Yeah,
B
they're
cells.
like a binding site for antigen.
Correct. And where are the binding sites on the monomers? Yeah, on those differential regions on the U.
On any of the 3.
All of them, yeah. That's what makes them all different. So, IgG, most abundant. 75 to 85%, we're going to see IgG circulating antibodies are. It is the main antibody, the most effective of the antibody classes. When we talk about a secondary immune response, late primary response, this is where we're going to see most of IgG produced by the cells. So who produces antibodies? Plasma cells, which were originally what?
T-cell?
No,
White blood cells?
B
Oh,
cells.
sorry, B-cell, yeah.
B cells. They're white blood cells, but remember we have two different types.
Yes,
We have T cells, we have B cells. Okay, when B cells are activated, and what's going to cause them to be activated? Those little Ig what's on the surface of their, IgDs on the surface of their cells. When they come in contact with a specific antigen that they can recognize, and what causes them to be able to recognize that specific antigen? Yeah, that differential site on the antibody monomer, okay? So that place, the antigen binding site, is different for all the different types of antibodies that you have. That's what makes it different. You have the ability to make, or actually you have in place right now, and this is kind of mind-blowing, you have cells in place that can recognize every single antigen that you will ever encounter in your lifetime, whether you encounter them or not. If you never encounter them, those cells will never become activated. They'll just kind of hang out in the lymph nodes, they'll reproduce themselves every once in a while, and they'll never become activated. But because of the fact that you can shuffle around and create different antigen binding sites on these different antibodies, you can fight any antigen that you ever come in contact with. You can always fight the war. It doesn't mean you'll win. Yes? Okay. So IgG, very important, very abundant, and important to remember that's the circulating guy. Can also cross the placenta and help with passive immunity. And then we have the IgE, also a monomer, and we see it attached to cells that are usually contain histamines. When they become activated, they release their histamines. Histamines are help with what? Yeah, inflammatory response, getting things to the area when they're needed. Secreted by plasma cells in skin, mucosa. We find a little bit in the plasma. And then when we talk about allergic reactions, our immune system pumps out quite a few of these IgE monomers. All right. You with me so far? So we have T cells, we have B cells, we have antigen-presenting cells, we have phagocytes, we have antibodies, we have cytotoxic cells, we have membrane attack complexes. All of these guys are part of an immune response. All of these guys are going to work together, band together, to help protect you from foreign substances. This diagram shows how several of these different cell types work together. When we talk about cells cloning themselves, they typically come from one cell. And in your textbook on page 783, they talk about monoclonal antibodies in clinical research tools. If I can find a cell that can recognize an antigen of interest, that can help to make antibodies against it and neutralize it, I can use that to then make detection kits. And I know that you've all come in contact with some sort of detection kit in your lifetime that was made from possibly this type of research. Anybody ever have a strep test? Yep. Anybody ever have a pregnancy test? Yay! Yay, May, yeah. So these kits are created and used all the time in clinical settings. And they're created using the principle of an immune response. So I used to do this for a living. We take an antigen of interest. One of the companies I worked for, we would extract antigens from the surface of cancer cells, breast cancer, cervical cancer, prostate cancer. And we take these antigens and we would inject them into mice. Now what's the mouse going to do when the antigen comes on board?
Make antibodies.
Do this, go through an immune response. Primary, secondary response. We'd inject the mouse several times, and we'd make lots and lots of circulating antibodies, wonderful IgG antibodies circulating in this mouse's system that would fight off these foreign substances. But where are the cells that produce these antibodies? In the lymph nodes or in the lymphoid organs. So after we injected a mouse several times with a specific antigen from the surface of those cancer cells, we'd go in and harvest the cells that we were interested in. Spleen was the best because there was tons and tons of cells in the spleen. And then we would go and find that cell, find the cells that were making the antibodies that we were interested in. Once we found the cell, and we did this with a chemical test where we actually looked at the antibodies that were produced by these cells, when we found the cell of interest, we would take that cell and do what with it? Make a whole lot more. If I make more cells, allow them to reproduce themselves and make lots and lots of antibodies, I can then harvest those antibodies and use them in our detection kits. So this company that was harvesting those cells that were made against proteins for breast cancer and cervical cancer, guess where their product was used? Yeah, when you went for a biopsy, say you suspected you might have breast cancer, you go for a biopsy. What are they doing? They take a chunk. They take a chunk, they take some of those cells, and they put them on a slide, and then they use the antibodies that I created in the lab from that one cell that I found, and it's called a monoclonal antibody because it came from one cell that made a whole bunch more. I can mark that antibody with some sort of dye so it jumps out at me on that tissue sample, like a fluorescent dye, for example. And I can see whether those antigens that I had gotten from the original breast cancer cell are on your cells.
So that's how you identify it's cancer or not?
That's one of the ways. Yeah, and that this company marketed, that's what they did. They made these detection kits for different types of cancers.
How about to use those antibodies as a treatment?
Actually, that is also in the works. Way back when, what we were trying to do way back when, and I know that they've succeeded in several ways of doing this, is encapsulating, say, chemotherapies, for example. Chemotherapy, really toxic drug, doesn't know the difference between healthy and normal cells, is going to kill everybody. But what if we could encapsulate that therapy and mark it so that it will only attack the cells that have those antigens on them? So that is also being worked on as well. That was one of the things our company worked on, is encapsulating treatments so that you could actually target only the cells that had those antigens. Yeah, it was just in the beginnings. I think we have had some successes. There was two companies I worked for. The company that I told you about, the monoclonal antibodies, that was a company called Matratech, and they were out of Belmont, Massachusetts. Because I moved to Maine. My husband's fault. Really? In Massachusetts? Yeah. I started out in the beginnings of Matratech way back when the fellow that developed the technology was working on what we call nuclear matrix proteins, which was groundbreaking at the time. So rather than looking at the proteins that are on the surface of the cell, he thought that it would be much more specific if we could look at proteins that were actually on the nuclear envelope. So he developed an extraction process where we were able to take those proteins and then make antibodies to those proteins. So that his detection kits actually target the nucleus of these cells, abnormal cells. So it was really neat research. But yeah, I know that my brother-in-law was from Mount Auburn Hospital in Cambridge, and he's actually worked with the fellow that developed Matratech, and they've actually used doing similar research. Then I moved on to a company called BASF Bio Research, and that was a larger company. That their main company was a company called Knoll Pharmaceuticals in Germany. And one of the things that's very tough to do in Germany is to do animal research. So they developed a division here in the United States, because there's tons more restrictions in Europe than there is here in the United States. No, I love mice, actually. I think they're very cute, but when you have to, I didn't unfortunately grow attached to them because I had to murder them on a regular basis, so, and do terrible things to them, like inject them with antigens and then go rip out their spleens and harvest their thymuses. And it's fine. Well, sometimes not the way we did it. Unfortunately, sometimes we had to mix our antigens with some nasty chemicals called adjuvants to stimulate more of an immune response in the animals. So it could be nasty. But then you'd go in and harvest the cells, and that was part of my job was to create the antibody producing cells in the animals, and then go and harvest them and make these monoclonal antibodies.
I would rather PD maybe chicken, but not mice.
Oh, mice are adorable. I think they're so cute. Chickens are nasty.
Millions of them.
I know. That's why they're so great to work with. And don't make me angry, because I can pull a thymus out in 3.5 seconds. That's what I tell my family. Don't make me mad. I can cervically dislocate you and pull out your thymus in no time.
Wow. And you're teaching at Central Maine.
Yeah, that's right.
Wow.
There's no research places around here, unless no, one of the most wonderful places, and we got most of our research animals from Jackson Labs, and they do wonderful immunology research there. Amazing place. I wish I could go to Jackson Labs. So if anybody wants, Bar Harbor. It's an absolutely beautiful place.
Isn't there one out of like Scarborough or Portland area, like Anterex or something like that?
What's it? Oh, why can't I think of the name of it? There's a couple of places in Portland, though.
That's my uncle.
Oh.
And was coming up with some sort of test for some type of prostate cancer.
Yeah, and a lot of those companies, that's their goal. They try to target a certain product that they can market for certain cancers, certain test kits, things like that.
Yeah.
Again, strep. And what they would do is take those antigens and inject them into rabbits, and they do the same thing. There's tons of different companies. Actually, I met a fellow who made antibodies for different companies in goats. So he actually didn't kill his goats. He would inject antigens into his goats and then harvest antibodies from their plasma. So he'd bleed them periodically and harvest the antibodies. And you can do that with a very specific column chemistry-like test, where you can extract the antibodies from liquids or humors and then release them into your product. So the guys that make those antibodies that you use for blood typing, it's a similar process. So they're going to elicit an immune response in some sort of creature by injecting, say, type A blood cells into them. Not enough to kill them, but so they'll create that immune response and make antibodies for them. Then they can harvest those antibodies and use them in products. So there's tons and tons of different companies that do that. So these are major types of T cells. Remember, all those guys that I just mentioned, sorry to get off track, but all those guys I just mentioned are going to work together in some way, shape, or form. B cells, T cells, plasma cells, all those guys. And one of the groups of cells that are going to help organize the whole immune response are T cells. So B cells, they're the business guys, yes? They're going to make the antibodies, they're going to make the memory cells, that's going to help neutralize those guys. But who helps to direct them? Who helps to tell them what they have to make antibodies to? And which cells have to come into play? That's the T cell's job. Now there's two different groups of T cells. There's a group of T cells called helper T cells. And there's a group of T cells called cytotoxic T cells. Both these T cells are going to help in an immune response. But the biggest player, the most important one, is the CD4 positive helper T cell. Now what the heck is CD4?
It's a protein that on the surface.
Correct. CD4 is a protein on the surface of this cell. That's how I can tell who it is. If I go under the microscope and I do a right stain and I do a white blood cell differential, can I tell who's who? Absolutely not. They're different. They're all different. They all have different jobs. They all play different roles. And it's based on the surface markers. If I had very specific stains that I could use to find that marker CD4, and I could probably make that in the lab for you too, I could tell the difference between a CD4 positive and a CD8 positive, which is what we call our cytotoxic T cells. So it's basically different proteins on the surface of these cells that tell the difference between one and another. MHC, that we talked about before, is a protein on the surface of cells that's going to help me present antigens in an immune response. MHC class 2 and MHC class 1 can be seen on the surface of antigen-presenting cells. And these cells are the guys that are going to help show everybody in an immune response what it is they have to go after. Remember we talked about antigen earlier?
Yeah.
What was it that was different about antigen? No, what was on an antigen that allowed me to distinguish what it was? What are those little antigenic determinants? Is that ringing a bell? Yeah, those little antigenic determinants. Remember that way back when in the diagram we saw the antigen and then we saw three little things sticking off and those were its different determinants? Well, those antigenic determinants are going to be presented to the cells in an immune response by antigen-presenting cells. The different MHCs are the place where I'm going to present those antigens.
On the surface, right?
Correct. So on the surface of CD4 positive T cells, we see MHC class 2. Again, MHC is a place where I'm going to display antigens that I'm under attack for. It's also my self-antigens. It's how other cells know who I am. It's how my immune system knows not to destroy my cells. You with me? MHC class 1 is on the surface of what? CD8 positive T cells. Again, cells are going to present antigens once they come and encounter them. These antigen-presenting cells are going to help activate cells in an immune response. So we see them there kind of halfway down the diagram, coming in contact with MHCs to help activate CD4 and CD8 positive cells. Again, just like the B cell, once I activate a cell, it's going to go into attack mode. And what is it going to do? It's going to break out, start to proliferate, make more of itself, and do one of two things. One, it's going to go fight the fight, or it's going to become a memory cell. And where am I going to find those memory cells? Yeah, in the lymphoid organs. Remember my little strange analogy before? I said one cell before I come in contact with any given antigen, but after I undergo an immune response, now instead of one cell, I have 10 cells or 20 cells. Those are the memory cells I make. So I make memory cells when B cells are activated, and I also make memory cells when T cells are activated as well. CD4 positive T cells are going to help all the other cells in an immune response know what it needs to do. I call them the cruise director of an immune response. Without CD4 positive T cells, my immune response is not going to be able to be organized enough to fight a given invader. And this is the problem with what nasty little virus? Yeah, HIV. Because they mess up CD4 positive T cells. Remember I told you that was the door that that virus used to get into that cell. When they mess up CD4 positive T cells, the whole organization of an immune response can't happen properly. So I succumb to things that I normally wouldn't when I'm in full-blown disease. So HIV versus AIDS. Okay? So, know the difference between CD4 and CD8 positive T cells. Understand that there's different MHC classes on these different cell types. You can see that on page 784 in your textbook. This table on page 786 helps us to see where I might find class 1 and class 2 MHC. And again, MHC self-antigens are important to present antigens to other cells in an immune response. Activation and differentiation of T cells, again, once those antigens are bound, once antigens are presented, we see that they start to proliferate. Co-stimulation, we can stimulate signals with other antigen-presenting cells, other proteins in an immune response. Proliferation, differentiation, that's important so that we can get enough cells on board to do what? Yeah, have a full-blown immune response and neutralize the invaders. Not only neutralize, but then get rid of the invaders as well. Yes?
Can I ask you, why, even though class 1 MHSC and class 2 has a different cells,
Are on different cells.
Yeah, there's a, the class 1 has all cells, pretty much they have this class 1 protein, and class 2 with only dendritic cell, macrophages, and B cells.
And
But
and CD4 positive T cells.
Yeah, but why in the clone selection they using not just a regular cell? They said that usually, even though it can be found on all nucleated cells, but it's actually it's using dendritic cell to activate T cell
CD4 positive T cells. Yep.
Why are they?
Well, what was CD4? How was CD4 positive T cells different than everybody else in an immune response? What did I say about that cell? What did CD4, had a special job. What was the special job of CD4 positive cells? Those are the helper cells. So these CD4 positive cells are sort of the main activation system. Once those guys get their direction, and they're going to get it from class 2 antigen presenting, right? They're going to set off the rest of an immune response. So they are extremely important in an immune response. Why? It's just because that's what we have as humans. We have two different classes. And what we see is those CD4 positive T cells are activated by that second MHC class.
But CD4 is a helper cell.
Correct.
It's also helping CD8
Correct.
to activate.
It helps everybody.
To activate B
Correct.
cells as well.
But remember, we need on every cell to be able to recognize it as self. So all cells need some sort of self antigens. So we see that all nucleated cells possess class 1, and class 2 have that special little job of helping to activate CD4 positive cells.
I don't have a question about activating CD4, but I have a question about CD8, activation of CD8.
Yep,
Because
class 1.
Class 1, yes.
Yep. And what do CD8 positive T cells do?
Cytotoxic.
They're cytotoxic. And what do those guys do?
They fight.
Yeah, they actually create chemicals, and we're going to talk about those in a minute, that can actually poke holes in things. So they have a different job. Even though they're both T cells, CD4 positive T cells are more involved in the coordination of an immune response,
Where they usually activate in the lymph organ as well?
wherever they come in contact with them.
Even it's in the skin or something?
Yep. It depends on where they are.
Oh, okay.
And usually it's in the lymphatic system. Okay, they're actually going to do their job in the periphery. Does that make sense?
Yes.
Okay. So, different chemicals, and we talked about these when we talked about the proliferation of white blood cells. Remember when we talked about cytokines and interleukins, when we talked about the proliferation of some of the different white blood cell types? They also mention those here as well. Hormone-like substances that are going to cause cells to do what they need to do in an immune response. Macrophages, stimulate them to do their thing, stimulate T cells, and those interleukins are going to help prevent other cells from becoming infected that we talked about in the beginning of the chapter as well. So we have two different types of T cells. We have helper T cells, CD4 positive T cells. Those again are the directors of an immune response. And you see them usually denoted as TH, T helper cell. B cells. And who's going to help activate B cells? T cells, exactly. So we see in this diagram, helper T cells help in an immune response, again, by getting other cells to understand what it is they need to fight. When we look at these diagrams, we can see that little red blob, whatever that little red blob is. And these are antigens that are going to help in an immune response. So, T helper cells in humoral immunity, T helper cells in cellular immunity. Cellular immunity, think CD8. CD8 are cytotoxic T cells. Those cells are actually going to cause problems to other cells. So, not only do we have to coordinate the cells that are going to help get rid of antigen-antibody complexes, we also have to help coordinate cells that are actually going to do some damage as well. So, activating CD8 cells is also the job of CD4 helper cells.
Yeah, my question was, now I can be clear. But you see there's a dendritic cell who's actually presenting antigen or
MHC,
1, right?
class 1 and 2. In this guy, he's got both.
Yes, I know, but the dendritic cell cannot be in the skin, for example.
Yeah, it can be in connective tissue. Usually, that's where we find it.
In the connective tissue, but not in the lymph organ, right?
Yep, we could see them there too.
So there and there.
Yep. What's lymphatic? What's lymph tissue?
Lymph tissue? What do you mean?
What is it?
Lymphoid organ?
What type of tissue is it?
Oh, it's a reticulum.
Reticular connective tissue,
Yeah.
right?
Yeah.
So, see?
Okay.
Yes.
Yes, for me.
So there's connective tissue. Your lymphatic system is connective tissue. Your dermis of your skin is connective. See what I mean? So you can find them,
So, okay, now
yes,
this is what
clear.
Yes.
So the nice thing about that one in that diagram is you can see MHC class 2 is going to present to who? What's its job? CD4 positive T cells. And MHC class 1 is going to present to who? CD8 positive T cells. So again, it's showing them what they need to do in an immune response. So there's different subsets of helper T cells that are going to help. Again, this is fairly new stuff, but helper 1 cells stimulate inflammation. Helper 2 cells defend against things like parasites in the body. Helper 17 cells initiate something called IL, interleukin 17, and that's also involved in an immune response, inflammatory response in an immune response as well. So the next thing they talk about is cytotoxic T cells. Now these are CD8 positive T cells. Have I lost pretty much all of you, or are we good? I told you this chapter was a yeah. Did you guys look at your interactive physiology? Yeah, you. I don't know. Depends on how much you pay me. Oh, I'm sorry. Yeah, we're not. I noticed that. I was like, rush, and I'm like, oh yeah, we're not behind. I might be nothing. Well, we'll see how far we get. We'll see how far we get.
Maria, can I offer something, honestly, because we talked to Katie, and can we stay here after class and just, if somebody has a question.
I have a lab to teach after class.
Yeah, can we stay? No, we.
Oh, you might be able to. There's a guy that usually comes in here after. I'm not sure, but he.
Not
Yeah.
for long, maybe for.
Yeah, if somebody comes and says I need the classroom, then yeah. But no, yeah, you can stay. Sure.
And maybe if somebody has a question, they can stay, and maybe we can together.
Yeah. Oh yeah.
Help each other to understand.
Yeah. Yeah, you can, as long as somebody doesn't need the classroom. We'll see. All right, so cytotoxic T cells then, CD8 positive T cells. They don't do the coordination thing. They're actually, when they become activated, what are they going to do? They're going to poke holes in other cells. So in this diagram on page 789 in your textbook, we see cytotoxic T cells and how they actually do their thing. Basically, they're going to produce chemicals very similar to what we saw complement doing, creating complexes that poke holes in things. Now, these guys make chemicals, you ready for it, called perforins. And those chemicals do what? Perforate. Exactly. They create similar pores to what we saw in that Mac attack that poke holes in cells. They also inject some nasty little chemicals called granzymes. I wonder who came up with these terms. Yep. And they are going to activate enzymes within cells and cause those cells that they're attacking to undergo apoptosis. What's apoptosis? Yeah, it's cell suicide. It's programmed cell death. And that's programmed into our cell cycle. Way back when you talked about mitosis and meiosis, remember all that wonderful stuff last semester? And there's different checkpoints a cell has to go through depending on what phase they're in. Remember G1 and G0? At some point, a cell says to itself, okay, I passed through this point, it's time for me to die. And that is apoptosis, programmed cell death. And it's all part of a cell's life cycle. These granzymes push a cell into apoptosis. So not only by producing perforins can I poke holes in things, but then I also inject nasty chemicals into the cell and cause it to kill itself. And that's how T cells work. So perforins and granzymes, the combination of the two, are going to cause cells to become destroyed. In this one, we see a lovely cytotoxic T-cell attacking a cancer cell. Now, not to scare the living daylights out of you or anything, but you are constantly making cancer cells. Do you know that? Why aren't we all sitting here with cancer? No, because these guys are constantly on surveillance. Cytotoxic T cells are constantly on surveillance. How do they know that that cell is to be killed?
Because they present.
Well, they see what on its surface? Something's not right. It's not self. It's not MHC1 or MHC2. I know that there's something wrong with that cell. These are non-specific. Yes? They become more specific when they get activated during an immune response. They actually go and attack a specific player. But they're constantly on surveillance in the body, attacking cells that don't fall in the that self category. So when they see cancer cells, and how do they know it's a cancer cell? What's different about a cancer cell than a normal cell?
The protein.
Yeah, the proteins on the surface of the cell. So these, again, can also fall into that non-specific category, where they're actually killing cells that aren't self.
I think when you hear the ones getting cancer, is that because their cells aren't mature enough to recognize these?
We can always fight the fight. We don't always win. So even though we have the ability to do this, sometimes cells can do what? Hide under the wire or stay out of the line of fire long enough to make a whole bunch of them. So that's when we get cancer, when these cells, our immune system, for some reason, haven't been able to destroy them at fast enough a rate, or they make it under the wire for long enough so that they can make a lot of themselves.
Is natural killer also destroy those cells?
Yeah, it's cancer cells, yes.
Yeah.
Yeah. And NK, natural killer cells, cytotoxic T cells, all fall into that same group.
Like, some cancers are hereditary. Is it, does it have anything to do with the fact that
Some of us tend to have a higher percentage of certain cancers based on our genetics.
does that have anything to do with like an issue with a genetic issue with T cells?
Not necessarily, no. And again, there's so many different types of cancers. We also have to take into account that our genetics are not the only thing that define the way our body is going to function. Environmental factors can either turn on or suppress certain parts of your genome for longer periods of time. So say I have a gene that might predispose me to breast cancer. Does that mean I'm going to get breast cancer? No. I have the gene. I have a higher percentage of maybe developing breast cancer, but environmental factors can either increase or decrease that gene's possibility of turning on. Does that make sense? So you can speed it up by exposing your body to chemicals that tend to cause cells to mutate at a faster rate. What do we call those chemicals? That cause cells to form cancers. It's cause cells to form cancer cells at a faster rate. Carcinogens, exactly. So we know that certain chemicals increase the chance that our cells are going to mutate and form cancer cells. We know smoking does that, yes? We know certain toxic chemicals do that, and that's what carcinogens are. So again, environment plays a role in it as well.
That's why it's cancer right now. That's
Well,
why
and
the
then
environment, chemicals in the atmosphere.
exactly, and that's what we have to remember as humans, is we might be putting stuff into our environment that we don't think is going to affect us, but we live in a bubble. We live in a petri dish, and the more we put out into our environment, it's eventually going to come back on us as well.
I did an essay, 7 pages about carbon dioxide. I'm sorry, not carbon dioxide.
Monoxide?
something that, the gas that we use in the car.
Yeah.
That's
Carbon dioxide.
Oh, right. So I did an essay about the influence of this carbon dioxide, not only for the cars, but for any,
In our environment,
in our
acid rain and all that.
environment, and how we can actually decrease the carbon dioxide for just simply switching like our lights bulb that we use in our home for different ones, and that it's another thing. There's a special, actually they actually
Fluorescent bulbs, the what do they call those long-lasting ones? I can't think of the
Yes,
name.
they last longer, so
Yeah.
you actually, save money, and also
Unfortunately, one of the downfalls to them is they have mercury in them, so which is also a toxin to our environment. Yeah, we got to figure out a better way to do all of this stuff. And as humans, we have to realize that we're affecting generations to come.
Do we need to know all of this?
Yeah. Oh, you don't need. What I want you to know is similar to what I had you know before. Interferons, alpha and gamma interferon, beta interferon. What are they doing for us? They're going to help cells, activate cells, help stimulate the synthesis and expression of class 1 and class 2 MHC. So interferons are going to help with immune responses in those ways. Interleukins, and these are some of the guys, and again, you don't have to memorize all the numbers or anything like that, but understand that interleukins are going to help stimulate cells in an immune response as well. There's a bunch of different ones, and they all play different roles in helping to secrete antibodies and the help of helper T cells to perform their interaction with other cells in an immune response. So that's pretty much it for that one. Now, the book also talks about organ transplants. So why can't I just grab my liver and give it to Mila and be done with it?
Thank you.
There you go, Mila. Have an extra liver, huh?
Your body won't recognize.
Exactly, because of all these cellular interactions in this chapter. Now you hopefully have a greater appreciation for how complex they actually are. All of the different cellular markers that you might possibly possess. Your MHC class 1 and your MHC class 2. Are my MHC the same as your MHC? No.
Maybe.
They may be. Maybe you can have some of my liver.
If you're identical twins.
Not necessarily. Not necessarily. Again, it's kind of like the lottery, if you think about it.
Have organ banks and stuff like that because you register and then they find a match
And when we find a match, what are we looking at?
one that your body won't immediately reject that has like the same kind of antibody.
Yeah, that's what I'm looking for. Very similar cellular markers. So the only ones that are going to have the same cellular markers are who? Yeah, the ones that have the same genetic information. And that would be identical twins. I might not even have similar cellular markers or enough similar cellular markers as my children. My children that I gave birth to. Because of the mix of genetics between father and mother. So, when we look at these cellular markers, all of these guys come into play. So it's not just blood type and blood type antigens, it's all the rest of these antigens we have to look at as well. So they talk about different types of transplants, and again, the autograft is the best. How come?
Because it's a part of your body. Maybe
Yeah,
tissue
comes from me.
transplant.
My tissue. That's the best. Isograph. And that's going to be from somebody who has the same genetics. So if you have an identical twin, you have some spare parts there. Yes? Not a fraternal twin, an identical twin. A fraternal twin is what? That's a totally different person. You just happen to be hanging out together and born at the same time, right? But you're a totally different genetic person.
Aren't there some cases where fraternal twins, one can be older than the other by like a month?
Yes. Mm-hmm. Yep.
You could deliver one baby and then deliver another baby in like another month.
Yeah, but usually you have to deliver them at the same time because the whole
Have to go
push
through
it
that
out process is,
Very rare.
it's rare.
So is it
Most of the time when you become pregnant and implantation has taken place, you don't ovulate. But that's, again, it happens. So it can, the next month, you can become impregnated with another offspring.
That's why sometimes when they do your ultrasound, they see one baby and then they see another 2 babies possibly in their ultrasound. My friend,
Does it happen to you?
No, not me.
Not you personally, but I
My
mean,
friend
you've
actually-
experienced that.
My friend just had twins on the 27th of September, my sister's neighbor, and she had a boy and a girl, and they told her she was having one baby, and then she ended up with 2. And she... Yeah.
Are they different? It was one much
They-
smaller than the
yeah,
other?
they said that one baby was developed at a different time
Mhm.
than the other baby. And
Yep.
it was crazy. I was like, really?
Yep.
Okay.
No, it happens. It can happen. And two different fathers could also be the fathers of your offspring as well.
I don't think that's
Craziness, right?
Yay!
Does that happen with, like, not the different father thing, but like fraternal twins, like, that are
You-
born, like they're born together, but
You ovulate two oocytes for some reason, and that happens every once in a while.
right, but is it more common that fraternal twins are actually like different, like they were conceived at,
No,
different
usually it's in the same
The same.
time period.
But
Yeah.
it's just
It's
one
unusual that
is
happens.
implanted at a different time than the
Yeah.
Is it very obvious when that happens?
Yeah.
Oh, okay, so they know, it's not just like,
Well, you can see developmentally, the other one is premature, where the other one is developmental age, so that 40-week age. So yeah, they can tell.
How come some, animal transportation or...
Animal, because you're animals.
No. It says that xeno or...
Xenograft.
Xenograft,
Yep,
that you can actually have a heart from bamboo.
yep.
if they do that.
Or heart valves from pigs.
Really?
Yep. Actually, one of the most common mitral valve replacement surgeries is pig valves.
My goodness.
So allograft would be from the same species, and xenograft would be from a different species. But again, what do we have to look at? Cellular markers. So we have very similar cellular markers to baboons. So our immune system isn't going to attack that tissue. But again, it's not just ABO. We have to look at all of these other cellular markers as well. Simplified summary of a primary immune response. So in this diagram, we're going to make it simple, right? Is your head ready to explode yet? But think of it as a coordinated dance. Who's the big dance coordinator? Which cells? Which T cells? Helper T cell. And then everybody else has to play their part in this coordinated dance. I like this diagram because we see everything tied together in this diagram. So something comes into the body, intruder. It could be a bacteria, it could be a virus, it could be a foreign cell, whatever it is. And there's going to be two main players that are going to help fight it off. We have adaptive immunity and innate immunity. What is innate immunity? It just knows it doesn't belong and I'm going to get rid of it. So we can have a barrier like skin, or we can have cells, and we've just talked about some of them, that know that doesn't belong, I'm going to kill it. Yep. Complement, interferon, cytotoxic cells can be that as well. Phagocytic cells can be that as well. But if they make it by those guys, that adaptive line comes into play. So notice, there's two groups in this diagram that are going to help with the whole immune response. First of all, we have those wonderful antigen-presenting cells, and they're going to help activate players in this dance, T cells. They help activate helper T cells, who also have a very close connection to the other side of the diagram. Who's on the other side of the diagram? B cells. Remember, those guys are important in adaptive immunity, specific immunity. I'm going to fight you specifically immunity in making those specific antibodies. But who's going to help coordinate proliferation of enough B cells to make enough antibodies to neutralize any given antigen? Helper T cells. Okay? So even though they can do a little bit on their own, they're not going to be able to do it efficiently and to the number that we need unless they get help from those helper T cells. Remember, the helper T cells are also going to play a role in helping to activate CD8 positive T cells. Those guys are the run around and poke holes in things T cells. And they're also going to specifically, in this case, of adaptive immunity, help destroy foreign substances.
And they actually will help to activate CD4 as well, right?
Well,
For co-stimulation.
Co-stimulation, yes.
They will help both.
Correct. And who's going to help co-stimulate? CD4 is going to help stimulate helper T cells. That's what co-stimulation means. Okay? So all of these guys play a very specific role in adaptive against specific immunity. Look at this diagram. It's going to help tie everybody together. Yes? I know, your head is ready to explode. But that's what's nice about this guy. If you can look at this guy and sort of explain all of the different players, where they come from, what is their role in an immune response, that'll help make it a little less overwhelming.
You know,
Yes?
as you gave us very, very great advice to put everything in one pretty much page. This is what I did for, all,
But doesn't that help bring it, when you take so much stuff and you bring it down into a couple of pages, it's going to help. And you studied the nervous system last semester, right? If I try to remember 7,000 different things in 7,000 different places, my head is going to explode. But if I bring everything down into a few pages, into one place, it's so much, you can review it and do that repetitive stuff that you need to make you remember.
I can
Okay.
honestly say the nervous system was way easier than ours.
And what it is, it's complex because there's so many different players coming in from different sides.
I'm so lost.
But try to look at that page 791 and see if you can understand everybody's role. This one ties everybody together. I like when they put everything on the same page. Okay? Now, this table is everybody we talked about. So this table is going to give you all of the players in the fight. Yes? So who do we have for cells? Cellular immunity, also part of adaptive immunity, also part of innate immunity. Some of these guys play a dual role, right? B cells, plasma cells, helper T cells, cytotoxic T cells. We have a couple of other guys in there called regulatory T cells, memory cells. Those are the guys that are going to be able to be on board to fight quicker in a secondary immune response. And antigen-presenting cells. These are the guys that are going to help show everybody exactly what they need to do in this immune response. So those are the cells. Then what else do we have? We have chemicals that are going to help us too. Antigens are the things that initiate an immune response. Remember, antigen is just that, a substance that can elicit an immune response. But not all antigens are going to do that, right? What antigens are not going to do that?
Self-antigen.
Yeah, it's self-antigens. And your cells actually have to go to school to become what? To know what a self-antigen is. Immunocompetent. To learn what self is. Yes? If I recognize you as foreign, I'm going to make antibodies against you. What do we call those? Immunoglobulins. And those substances made very specific to the antigens in question are going to help neutralize them so some of these other players can come in. Then we also have perforins and granzymes, poke holes and things and cause cells to self-destruct. We also have complement. What's that going to do? Poke holes and things, right? And cytokines, they're kind of the chemical messengers. They're going to help draw cells to the area. So this table here is going to help tell us all the players, and then we can then incorporate these players and what their job is in an immune response into the diagram before. Yes? So where do they play their role in this whole scenario? All right, you ready to explode yet? Homeostatic imbalances of immunity. So the immune system doesn't always work like that fine-oiled dance that we just talked about. Sometimes signals get mixed up. Sometimes cells flunk kindergarten and we don't kill them. They get out there. They start attacking themselves. Deficiencies in the immune system and this immune response are defined as immunodeficiencies. Huge, huge, broad term, right? Because there can be so many different immunodeficiencies. There's a my brain just went dead. Virus that can mess up this wonderful fine-tuned oil machine we've talked about several times already in this chapter, HIV. So HIV is a virus that does what? Which part of the immune system? Which T cells? The CD4 positive helper T cell. Remember, that's my cruise director. If my cruise director doesn't know what to do, nobody else knows what to do. So that's what HIV does. And it messes with those helper T cells, the CD4 positive T cells. I can no longer coordinate a systematic immune response. If I can't do that, I can't fight off foreign invaders. Full-blown AIDS is acquired immune deficiency syndrome. And that's when the virus has made it so severe that I can't react to very simple antigens in my environment. Epidemiology since 1981. Wow, that just made me feel really old. Because, yeah, when I was back in the clinical lab way back when, we didn't do things like wear gloves. We used to eat in the lab. We used to smoke in the lab. Back in the day.
You smoked?
I did. I'm much more knowledgeable now. But it was in those early 80s when we even discovered the epidemiology of HIV and AIDS. Treatment and prevention is going to help. There's no cure. Viruses hide inside cells. They have a nice little hiding place. You don't typically attack your own cells, right? Yeah. So that's why viruses are so nasty. But they talk about some of the different therapies. And again, to help prolong that immune system coordination. And that's what some of the therapies help to do. Another category of diseases, immunodeficiencies, they talk about is something called autoimmune disease. And this is where what? The breakdown is you can't recognize self. So you start attacking your cells of this immune response will start attacking your own cells. There's a whole bunch of diseases in this category. They list just a few. Rheumatoid arthritis, you destroy your own joints. Myasthenia gravis picks on nerves and skeletal muscles. Multiple sclerosis, you start attacking your own myelin sheath. Why is that a problem?
Because the action potential or nerve impulse will not...
Right, doesn't make it to the end. Exactly. So when you talked about nerve impulses, your myelin sheath was an insulator, and that allowed nerve impulses to happen. When stuff sort of leaks out because the myelin sheath isn't effective, the action potential doesn't necessarily make it to the end of the neuron, and that affects nerve impulses. Type 1 diabetes, insulin-dependent diabetes, systemic lupus erythematosus can affect different organs like the kidneys, heart.
I never heard about this. Systemic lupus?
It's an autoimmune disease, and it again attacks different organ systems. My sister has lupus. My oldest sister has lupus. Lungs, kidneys, skin. And she's got to be careful. Can't overdo it too much, which she never pays attention to, so she overdoes it all the time. But it can affect those different systems. And glomerulonephritis, which it affects the kidneys and renal function.
How much of these do you want us to know?
Just a general definition.
Of all of those and autoimmune?
Yeah. Be able to recognize them as autoimmune diseases. So we block some of the different chemical messages and treatments for autoimmune diseases. And they talk a little bit about that under treatment of autoimmune diseases. Self-tolerance, failure for self-tolerance can cause problems with autoimmune diseases. And remember that positive and negative feedback that we went through in the thymus can cause problems if it doesn't undergo properly. So, hypersensitivities, immediate, subacute. Some of you might be going through some of these things now during allergy season. This is where we create complexes that can cause problems by causing more inflammatory responses to happen. More, what's the word I want?
Locally?
Yes, thank you. More localized inflammatory responses, hypersensitivities. Sometimes different detergents. Is anybody like this? You can't change your clothes soap because you'll cause your skin to react adversely to that particular substance.
Why some people has this and some people just don't have any allergy at all? And
I know, and it's because of the way your immune cells, it's our different, I can't give you, there's no pat answer to that. It's just the way your body reacts. Think about your body and the complexity of your system.
I never had allergy before I moved to Maine. Never had any like sneezing nose.
I never had allergies till I was 21.
When I moved to Maine, just last year, I started to feel the allergy. Not even allergy, I think like it's a hypersensitivity. And I don't know even from what. I think like maybe something growing. And I had it before.
And our systems change too. I never had, example, never had allergies, seasonal allergies, until I hit my early 20s. Of course, I developed seasonal allergies during the same season I had to take a class called Vascular Plant Taxonomy, where we went out into the fields and identified ragweed and stuff like that. It was a blast. So your system changes, your body changes.
But that could have also been because then you were more exposed to it and spending that amount of time out in that.
It could have been, or I'd been exposed to those things before and never reacted to them. We also talk in this chapter about haptens. We all have different proteins. They're called incomplete allergens. Things like poison ivy. So who in this class is allergic? I'm wicked allergic to poison ivy.
I've never had it, ever.
Never had, because why?
You have antibodies?
No, this is all in the hypersensitivity thing. You don't have the proper proteins in your body to hook up with the oils in the ivy leaves to create a complete antigen.
So I'm lacking.
So you're good. That's a good lacking. Yes?
Yeah, that's good.
Because you never create that complete.
Sometimes I've been tempted to like just walk through it on purpose to see if I can get it.
Well, honest to goodness. And then the more you're exposed, in this case, to those allergens, the more proteins you make, the more You
Yeah,
react to them.
It's like opposite reaction.
Exactly. So,
We used to go to a friend's house, she had a long, thick cat, and I'd go in, all of a sudden my nose would get stuffy, my eyes were watered, and I'd have a hard time sitting my throat.
yeah, because you'd experience a lot of what you see in this diagram here on page 795.
Like,
No, so when you go for shots, what do they do? When you go for allergy shots, what do they do? Exactly.
They would
They're exposing you in very low doses to some of the things that you react to and causing it to become more normal in your body.
I have a friend that I work with that is going through immunotherapy, and like she's posted pictures of like her back after she goes for it, and it's like a grid of hives.
Nasty. Well, that's what they're doing is they're trying to figure out what combination they should give her. So they're trying to see how reactive she is to certain things. Because when you go for allergy shots, they'll give you combinations of some of the different things that you react to. And I honestly, after working in immunology for so many years, I think when our bodies are in hypersensitive mode, we're going to react to everything.
Yeah. So it's a
You
certain,
know,
I think it's a certain period of
exactly.
time. It can just end any time.
Yeah, it could.
Good. What's the difference between type 1, type 2, type 3, and type 4?
Well, there are different players in an immune response are going to be involved. So type 2 cytotoxic T-cells are going to be involved. Type 3 hypersensitivities, different players involved. Neutrophils, for example, are involved in type 3. No.
Because it's a little
No,
complicated.
I'm not, yeah, I'm going to skip that part
She
because...
wants to know about hypersensitivities.
So just understand that hypersensitivities involve basically what we see in figure 21.21 on page 795. So plasma cells, those mast cells that contain histamines are going to become stimulated and over-release those histamines to cause an overabundance of an inflammatory response throughout the system. So histamines can cause blood vessels to dilate, become more leaky, and sort of be a never-ending cycle of fluid release during a hypersensitive moment. That can cause big problems if the hypersensitivity is more systemic and it hits your respiratory system. So a lot of these hypersensitivities, sometimes people can succumb to them if they have an allergic hypersensitivity by not allowing them to breathe. It attacks their respiratory passages and constricts their bronchioles.
What's the difference between hypersensitivity and allergy?
Allergies are hypersensitivities.
It's the same thing,
Yes,
right?
your body's reacting that way.
But they're referring to allergy when it's in severe or acute.
Right,
form.
and they call it allergens when it's more severe and acute, yeah.
When it's causing
The-
anaphylactic shock.
Correct. And that's going to be a severe, exactly. So we can have allergic reactions to lots of different things. I know how many people have walked into Bath and Body Works and during different times of the season, and you're like, get me out of here, because the scents in there are too much, right? Guys especially, my husband hates that, so he's like, I'm not going in there. And he keeps throwing away, and I'm going to beat him like a stepchild. He keeps throwing away. I buy those nice little plug-in things with the little things, and he keeps throwing them away. And I keep buying more.
It's
So.
like they can't stand going in those places, but they don't mind, the nice smells that you pick out. Like if you smell good, they'll certainly have no problem
Yes.
mentioning that.
All right, my friends.
Can I ask you a couple of questions regarding our possible essay question? How would you like us to?
I gave you those, right?
There's one of want you to draw and label electrocardiogram. Would you like us to draw like exact?
Yep,
Like,
PQRST.
No. I understand, but how would you like us to draw? Like
With a-
all,
No, just P. No. You don't have to
all
get
the
crazy.
seconds and
No. PQRST.
Yeah.
That's it.
That's all.
Yep, and then what each part
Do we need
means.
to know the intervals, like the P through R
Yeah,
interval?
there's
You're going to do that in lab.
Okay, so we don't need to know that for the test. And as far as explaining like what P, Q, R, and S, like if we do like, if like atrial depolarization, is that a clear enough answer?
Good for me.
Okay.
And then QRS would be what? Depolarize.
Contractional depolarization.
And then,
P is.
no, what else happens during QRS?
It's atrial repolarization.
Correct. So both of those things happen during QRS.
Okay.
And then T is going to be?
Ventricle repolarization.
Correct.
And another question, what exactly would you like to know, us to know about cardiac in-string conduction system, pathway, and action potential? Would you like us to know just if it go from CA node to AV node, or would you like us to know or to describe action potential first?
No, you don't have to
potential, and then go to that?
Nope.
Okay, just the process of conduction.
Correct. And the pathway of conduction.
Yeah.
So
And
I'm
that
trying to
starts
find
with
the,
that, right? Like where is,
Yeah.
where is happening?
so if you look at, and I'm trying to find the diagram for you, muscle fiber, anatomy. Oh, it's on page 675, figure 18.15. So that basically is the conduction system and action potential succession during a heartbeat.
Okay.
So it starts, it's page 675, figure 18.15. So if you have an older book, it's that picture of the heart with the little, whatchamacallits.
One last question. It's page 713, okay? And this is called, the section of this chapter called blood flow in special areas.
Mhm.
There is a skeletal muscles, brain, skin, lungs, and heart. What exactly? Because there's no like different points. It's more like
Just understand that when, and you don't, I'm not going to make you know numbers, I'm not going to make you know any of that, but understand that total blood flow rest versus exercise, what's going to be different? Things that stay the same, brain, that doesn't change. Whether you're at rest or exercise, your brain's going to get what it's going to get no matter what. What we start to see change is different blood flow to skeletal muscles. Because they're in use, we're going to see skeletal muscle gets more blood flow during exercise versus rest. So just understand that, and again, you don't have to know the numbers, you don't have to memorize that, but understand that blood flow changes basically based on need, but it's never going to change for your brain. So that's what I want you.
Exercise will increase the blood
Sort of
flow to skeletal
redistribute,
muscles.
exactly.
All right.
Yes? Yes? We good? We're going to get 100? 105?
On Tuesday, right?
No, Thursday.
Thursday.
What? I thought you were.
Yeah, but you know what happens? If I give you your test on Tuesday, then I'm going to start with the respiratory system on Thursday.
Yeah, that's already like mismatched.
No, I think
Yeah.
it will confuse us even more, guys, because if you realize that we have to do respiratory, then
So what do you think? I'll give you a vote. I'll give you a vote. All right, who wants their test on Thursday? Now be realistic with yourself. What are you going to do if I give you all that extra time?
I have to work all day my, you know what, off. Eric has
Are you going to though?
Yes.
I have to work tomorrow.
okay,
Yeah.
I don't have to work tomorrow.
and just follow the.
I just feel like there's,
To be
because I've done
completely
reading and
honest-
I've been doing mastering and I've done, I went in to see Dr. C today, I have done so many tests, I've been
So
taking out
by
cognitive
letting me,
And this new stuff, I need to, I'm not, in one day, I'm not gonna be able to focus on this and finish what I've already focused on to be ready for the test. There's absolutely no way. I
Okay.
honestly don't even wanna come to class. Something catastrophic has to happen to me tonight or tomorrow, because I can't be here Thursday.
I don't want you to do that. All right, well, listen.
A review in class on Thursday.
No, that's the deal. I'm not going to do that. So here's the deal. If I give you your test on Tuesday of next week, I am going to press on with new material.
Yeah, and then you
That's the
mess up
deal.
the respiratory
No, you're not going to mess it up, but I mean, that's the deal. I'm not going to take a whole entire class to review stuff.
I'm okay with that. I can handle that because I can just tune out if I have to and just watch the video later.
I think the thing is, because Maria already told us that test will be on Thursday.
Yeah, I did.
You guys-
But I will. I mean, I'll let you do that. But that's the deal. So I'll take another vote. If I do that, I'm going to go on with new material. So I'll take a vote again. Who wants the test Tuesday of next week?
Tuesday next week.
Who wants it Thursday? All right. Here's the deal. If you guys are hell-bent on taking that test on Thursday, you can come, and I'll give it to you before if you really want to take it Thursday. You can't do it during class period because I'm going to be lecturing you during class period. So that's the deal. I'll give the test to the class on Tuesday of next week. New material on Thursday. Okay? If you really want to take the exam on Thursday, actually, if you even want to take it tomorrow, let me know.
What time till do you work tomorrow?
I'm in my office tomorrow from 8:30 until just before 2 o'clock.